Hello dear students. Today we are going to discuss the second part of the chapter Principles of Inheritance and Variation. So in this session we are discussing some very important topics such as linkage and recombination, determination of sex, pedigree analysis, etc. The first prime point is linkage. Linkage means the physical association of genes in a chromosome. For example, this is one chromosome. The first gene may be here, the second gene may be here, third gene may be here, fourth one here, so that the chromosome, in a chromosome, the genes are arranged at different locations. The particular location of a gene in a chromosome is designated as locus. So, when the genes are linearly arranged in a chromosome, we can see that some genes are nearly located with respect to each other, and some genes may be distantly located to each other. Anyway, the physical association of various genes in a particular chromosome is called linkage. So this is chromosome number 1, and in the chromosome number 1 so many genes are there, and all the genes are considered to be linked together only because they are present in the same chromosome. Likewise, there is another chromosome called chromosome number 2. Here may be some other genes. Okay. But we cannot say that the genes in the first chromosome and the genes in the second chromosome are linked together. They are not in the linkage group. So all the genes in the chromosome 2 will be linked in a single linkage group, and all the genes in the chromosome number 1 will be also form a linkage group. That means the physical association of genes in a particular chromosome is called a linkage. So when two genes remain in the same chromosome, generation after generation, they are said to be in linkage. So here you can see that this is a chromosome. There is a gene called A, a gene called B, a gene called C. So here A and B are located very much near to each other. So these two genes will be remaining together generation after generation. So how the genes are get separated from each other due to a phenomenon called crossing over. So here you can see that this is one chromosome, this is another chromosome. At the type of crossing over, these two chromosomes will cross with each other, and here you can see that there is a capital A and small a, here capital B and small b, here capital C and small c. Okay. So here will be capital A, here will be capital B, here will be capital C. Here small a, small b, small c. So after their separation, they will break here. After separation, you can see that here comes capital A, capital B and small c. Here comes small a, small b, but capital C. Okay, because crossing over occurs here. So even after crossing over, capital A and capital B remain together, but capital A and capital C become separated. Small capital B and capital C become separated. Okay. So the tendency of two genes to be inherited together, the relationship is called a linkage. So for a strong linkage, a stronger linkage, the genes must be very much nearly located. When the distance increases, the chance of separation also increases. Linkage leads to parental combination of characters. That means capital A, capital B in the parent, capital A, capital B in the offspring also. When there is linkage, there will not be any independent assortment. So different combinations are non-parental combinations are not obtaining, only parental combination. Linkage is possible when two genes are nearly located in a chromosome. Linkage never occurs for two genes that are present in two different chromosomes. So, for linkage to occur, the genes must be present in the same chromosome. Moreover, the genes must be nearly located. Okay. When the genes are separating, that phenomenon is called recombination, that we can see in the next topic. Here comes the next prime point, that is recombination. Recombination is just opposite of what we have studied in the previous session. Linkage means the two genes remain together. Recombination means the two genes get separated from each other. When two genes present in the same chromosome get separated in successive generation, it is recombination. We already studied that. What is the reason for recombination? Recombination is crossing over, isn't it? The parental chromosomes will cross with each other and exchange their parts. As a result, the distantly located genes become separated. Recombination leads to non-parental combination. When there is linkage, the same characters combination of the parent will be sustained in the young ones. But when there is recombination, new and new combinations will be appearing. Non-parental combinations will be appearing. So when there is recombination, there will be independent assortment. Only by independent assortment, non-parental possibilities are there. Recombination is possible when two genes are distantly located. So suppose there is a chromosome, capital A, capital B, and capital C. So capital A and capital B, since they are nearly located, when crossing over occurs in this position or in this position or in this position or in this position, the capital A and capital B remain together. But any of these recombination crossing over will separate capital A and capital B from capital C. Okay. So when genes are nearly located, there is a highest possibility of linkage. When the genes are distantly located, there is a highest possibility of recombination. Okay. Now we are moving on to a very important topic regarding your examination point of view. That is the mechanism of sex determination, chromosomal mechanism of sex determination. You might have studied that there are two types of chromosomes, namely autosomes. Autosomes, otherwise called somatic chromosomes, and allosomes, otherwise called sex chromosomes. What is the difference? In autosomes, all the genes are controlling our normal non-sexual metabolic functions. Digestion, respiration, circulation, locomotion, sensation, etc. But in allosomes are chromosomes which carry genes which are controlling our reproductive activities, sexuality of the person, isn't it? So sexual genes are present in those chromosomes. That chromosomes are called sex chromosome or allosomes. And non-sexual genes are present in those chromosomes. That chromosomes are called autosomes or somatic chromosomes. So here, in the case of grasshopper, in grasshoppers, there is a peculiar type of sex determination called XXXO type. What is that? In grasshopper males, autosomal pairs may be there. In addition to autosome, there will be only one type of sex chromosome, only one X chromosome. The other part is 0. But in females, autosomal pairs are there. In addition to autosome, 2 X chromosomes are there. That means in male, only 1 X chromosome. In female, 2 X chromosome. 1 X chromosome is absent. That is represented as the XO. So here, male will produce 2 types of sperms. One sperm containing autosome plus X. Another sperm containing autosome plus 0, no sex chromosomes. And the female will produce 2 types of ova. One ovum consists of autosome plus X. Another ovum also consists of autosome plus X. Okay, that means the female produces only one type of gamete, male produces 2 types of gamete. So the males are considered to be heterogametic. The females are considered to be homogametic. When the ovum, capital A, autosome plus X, fuses with the sperm of the first kind, a female young one will be produced. When the ovum is fused with the sperm of the second type, a male individual will be produced. That means the sex of the fetus, sex of the baby, will be determined not by the type of the ovum, but by the type of the sperm. Okay? So females having 2 X chromosomes in addition to autosomes. Males having only 1 X chromosome in addition to autosomes. So females are homogametic. Why females are homogametic? They produce only one type of gamete. And males are heterogametic because 2 types of gamete sperms are produced. So the sex of the baby is always determined by the heterogametic parent. Here the males are heterogametic, so the sex of the baby is determined by the type of the sperm or the type of the male gamete. Okay. Now the next prime point is regarding the sex determination in human beings. In human beings, there is XX, XY type of determination. So here, human beings are having 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosome, allosome. So in males, we have 22 pairs of autosomes. That means 44 autosome plus 1 X chromosome and 1 Y chromosome. In females, 22 pairs of autosome, 44 autosome plus 2 X chromosome. So here also the male will produce 2 types of sperm. 2 types of sperms are there. That means 22 autosome plus X, 22 autosome plus Y. An X containing sperm and a Y containing sperm. But all the ova produced will be having 22 autosome plus X. Only one type of ova. That means the female is homogametic, but the males are heterogametic. When the X containing sperm fuses with the ovum, a female young one will be produced. When the Y containing sperm fuses with the ovum, a male child will be born. That means the sex of the baby here also determined by the heterogametic parent, that is the male parent. Okay. The females have 2 X chromosomes in addition to autosomes. Males have only 1 X chromosome and 1 Y chromosome in addition to autosome. So the females are homogametic. That means producing only one kind of ovum. Males are heterogametic, producing 2 kinds of sperm, which are the X containing sperm and Y containing sperm. The sex of the baby is determined by male parent. Why male parent? Because heterogametic parent. Now the next point, another type of sex determination is present in birds. That means ZW type. This one is different from the previous ones. Here, male birds, in addition to autosome, has autosomal pairs, has 2 Z chromosomes. But in females, in addition to the autosomal pairs, 1 Z chromosome and 1 W chromosome. That means male will produce only one type of sperm. That means autosome plus Z. But the female will produce 2 types of ovum. The female will produce 2 types of ovum. Ovum with the autosome plus Z, ovum with the autosome plus W. So, if the sperm fuses with the Z ovum, a male will be produced. If the sperm fuses with the W ovum, a female will be produced. Here, the female is considered to be heterogametic, as 2 types of ova are produced, whereas the male is considered to be homogametic. We already studied that the sex of the baby is determined by the heterogametic parent. So, in the case of birds, the sex of the baby is actually influenced by the female parent. The males have 2 Z chromosomes in addition to autosomes. The females have 1 Z chromosome and 1 W chromosome. So males are homogametic, females are heterogametic. Sex of the baby is determined by the female parent. Okay. Then there is another type of sex determination in the case of honeybees. This is not based on the type of chromosome, but based on the number of chromosome. That means the number of chromosome is represented as ploidy. Here, all the females are diploid, having 2n number of chromosomes, and all the males are haploid. So haploidy leads to male condition and diploidy leads to female condition. So how this happens? The male organism is haploid, produces haploid sperms by mitosis. The diploid female organism produces ovum, haploid ovum, by meiosis. So the spermatogenesis is by mitosis. Oogenesis is by meiosis. When the sperm and ovum fuses together by a process of fertilization, a diploid zygote will be produced, and this zygote develops into females, because diploid young ones are always females. But in honeybees, there is another mechanism called the ovum will develop without fertilization. Ovum can develop without fertilization. That process is called parthenogenesis. So by parthenogenesis, a young one will be produced. The young one will be haploid. The haploid young ones will always become males. Right here. That means in the case of honeybees, the males are produced by parthenogenesis and the females are produced by fertilization. So the sex of the baby is determined by the type of reproductive mechanism. And diploid young ones will always be females. The females are called queens, and the haploid males are called drones. So in honeybees, the females are diploid individuals, males are haploid individuals. The females may be queen or worker. Workers are sterile, not reproducing, but queens are reproducing. Males are drones. Queens and drones are fertile, but workers are sterile. Females are produced from fertilized eggs. Fertilized eggs, hence they will be diploid. Males are produced from unfertilized egg. The development of unfertilized egg is called parthenogenesis, hence they will be haploid. So there is another. So we already studied 3 different types of sex determination: XXXO type, XXXY type, ZZ, ZW type. And this is the 4th type, that is called haploid diploid mechanism. Okay. The last prime point in this session regarding pedigree analysis. Pedigree analysis is a pictorial representation of inheritance of genes in a family. So there are some symbols used here. The male organism is used as a square. A female organism is represented as circle. A male organism and a female organism is connected by a line that called marriage. A male organism and a female organism are connected like this. So brother and sisters. This the first one is husband and wife. Here it is brother and sister. That means this line denotes two offsprings. This is a son and this will be a daughter. Okay, these are the preliminary symbols. So here all the members of one generation are represented in one line. So this will be the first generation, the second generation, the third generation, and the fourth generation. So 4 generations are included in this pedigree. So in the first generation only 2 members, 1 and 1 2. So they have how many children? 3 children. The first child will be a son. The second child also is a son. Third child is a daughter. Okay. So this is the first son's of family. He has 2 daughters. So this is the husband of the first daughter and this is the grandson. The second son, the wife of the second son, first one son, second one daughter, the husband of the daughter and granddaughter. Then the third daughter, the husband of the daughter, only one daughter and the husband of the daughter. Okay, this is how the relationships are introduced in a pedigree. And by using this pedigree, we can represent the inheritance of genes, whether the dominant gene or a recessive allele will be inherited like that. That is another thing. So in this case, how a pedigree analysis is represented. By studying the pedigree, we can analyze whether it is an autosomal character, allosomal character, that means sex-linked character, then a dominant character, recessive character, etc., etc. Suppose if a person is possessing a character, that is represented like this. If a male is possessing a character, it will be black. Suppose if a female is possessing that character, like this. If a female is a sex-linked carrier, it's represented like this. Okay, so these are some other ways to represent how the inheritance of characters are represented in a pedigree manner. But here we are introducing the pedigree analysis, how to describe a family, how to represent the family relationships in a family in the form of a diagram. Okay, this is how the pedigree analysis will be drawn. Now, for example, there is a sample pedigree, a father, mother, and a son is having a particular character. For example, blue color. So, blue eye color. The brown eye color is the dominant one. It may be capital B, or capital B, small b. Blue eye color is recessive, small b, small b. Here you can see that the son is with a blue eye. That means the genotype will be small b, small b. But both of the parents are having brown eyes. So what may be the genotype of the parents? Here, how the small b condition appeared in the young one, the son. One small b from father and one small b from mother. Okay, then only the child will be small b, small b. So, but the father is supposed to be brown. A brown with the small b means capital B, small b. The mother is also having brown eyes. A brown eye means capital B, small b. Okay, so this is how we can identify the genotype of a person by using the pedigree analysis. Okay.